University of Burma, Japan. I will be chair in the morning session. The first speaker is Professor Aizawa. The title of his talk is Development of Novel Expression Systems for NMR Structural Analysis of Antimicrobial Peptides. So, Aizawa, please.
Thank you, Mr. Sensei. Okay, today I'm going to talk about our study about development of novel expression system for NMR structure analysis of antimicrobial peptides. Okay, first I'm going to talk about antimicrobial peptide, AMPs. AMPs discovered from variety of animals and plant species. And AMPs produced in many tissues and cell types. They are usually cationic small peptide, and they are classified by structure types. For example, linear alpha heteropeptides is one of the most famous type of antimicrobial peptides, and cecropin from insect and manganine from frog is one of the most famous family of that kind of peptide. And cationic, and other kind of AMPs contain cysteine and form disulfide bonds. For example, vertebrates defensin and insect defensin and plant defensin is also famous family of that kind of antimicrobial peptides. Okay, next I'm going to talk about recent topics about studies of AMPs. It is believed that AMPs kill microbes mainly by membrane targeting mechanism. So it is very important thing to analyze how AMP kill or slow the growth of microbes. So it is very important for us to analyze membrane binding structure of AMPs, and it is believed that AMPs form organometallic structure in membrane. So it is very important for us to analyze the membrane binding organization structure of AMPs. So NMR is very useful technique for analysis of this kind of organization. And other recent topics about the study of AMP is AMP interact with other intracellular molecules to exert their activity. This figure, this scheme represents that kind of interaction. And in order to study that kind of interaction, NMR is also very useful technique. Okay, so I'm going to talk about strategies for producing AMPs for NMR studies. As solid phase synthesis is one of the promising candidates for prepare the antimicrobial peptides, because antimicrobial peptides is very small and easy to synthesize chemically, but it is very difficult to use solid phase synthesis to prepare isotope-labeled samples. So recombinant technique by using microorganisms is very useful to prepare isotope-labeled sample for NMR analysis. Okay, that is direct expression is very simple, but it is difficult to prepare antimicrobial peptide by using microbes because degradation and toxicity of antimicrobial peptide is very important problem for producing that kind of small peptide. So overexpression as an inclusion body is very useful technique for producing AMPs. Okay, this is a good sample of toxicity of AMPs to the host cell. This is an example of direct overexpression of a certain AMP in E. coli cell as host. In this example, after RPTZ induction, yes, cell rises, and yes, this is a picture of the cell after induction. You can see clearly cell rises by the toxicity of antimicrobial peptide. So toxicity to host cell is a very crucial problem for producing antimicrobial peptide. So it is very useful technique for formation of inactive inclusion body for preparing the antimicrobial peptide. You know, inclusion body, IV, is inactive aggregates of protein, and it's inactive and protease tolerant. And other merit for using inclusion body is it's very easy for recovering from the host cell. However, the mechanism of formation of inclusion body is not clearly understood. And in the case of AMPs, it is difficult to produce inclusion body because small peptides often do not form inclusion body. So fusion protein technique and tandem expression technique are most promising candidate for producing that kind of small peptides.